What do you think would happen if you wrote down 18,000 random A, T, C's, and G's? And then you took that sequence of 18,000 random A, T, C's, and G's and you synthesized it. And then you took that synthesized sequence and you placed it inside of an organism. What would you expect would happen to those 18,000 base pairs? Would they be able to do absolutely nothing? Would they be integrated into the host's genome and operations? Would they be expressed? Would they wind up into a chromosome? Because a chromosome is more than just DNA. It's also DNA plus proteins. I've asked a similar question. Can random DNA sequences make things? In which I took 5,000 random base pairs and I assessed whether those base pairs could be transcribed and even potentially translated if you put them into an organism. But now it's been done. It's really been done. In this case, 18,000 base pairs have been synthesized, placed into a yeast cell, and then observations. What happened? What happened to those 18,000 base pairs? Let's explore that question coming right up. All right, the paper we're going to take a look at is published in the journal Molecular Cell. It's by a group of individuals from Harvard University, and it was just published just a couple weeks ago. Title: Functional Analysis of a Random Sequence Chromosome Reveals a High Level and the Molecular Nature of Transcriptional Noise in Yeast Cells. Yay! Sounds like a real winner, doesn't it? Really, it's interesting. It really is quite interesting. And in this case, I'm not going to delve into a lot of details. I'm not even going to show really many figures, or maybe I won't show any figures from this paper. They're very complicated, difficult to describe. I'm not sure I fully understand all the figures, but I do get the main point of this paper, and that's what I want to communicate to you. So let's just scroll down here, and we're taking a look at what's called the graphical abstract before we look at the full abstract, the verbal abstract. And what they're showing here is you have a typical yeast cell, right? That's the organism we're interested in here. Yeast cells are eukaryotes. Eukaryotes have a nucleus, just like you and I have a nucleus. And inside that nucleus, that's where we maintain our genetic information, right? Our genome. And so the nucleus includes a number of different chromosomes, and it does in yeast. They have a bunch of different chromosomes. And what they've done is they have created a random DNA sequence artificial chromosome. They've constructed a chromosome. And really briefly, what they've done is they've taken 18,000 base pairs of randomly generated sequence, which they then have synthesized. And that's actually an interesting process in itself, but we don't need to go into that. They end up with 18,000 base pairs of DNA. They put two caps on the end, which are typical chromosome ends. All right, they have the typical profile of sequence that will be recognized by an organism. And then they insert this artificial chromosome into the yeast cell, and then they watch and see what happens, right? Will this chromosome, or will this piece of DNA turn into a chromosome? Now, I need to explain that. DNA is a portion of a chromosome, but DNA itself is not a whole chromosome. You often may think that, oh, well, I have a bunch of chromosomes, that's strings of DNA. Yes, it's strings of DNA, but it's strings of DNA that are wound around proteins. You put the proteins and the DNA together, and that's what we call a chromosome. A chromosome. All right, so that's the first thing. They're just indicating they've made a cell with an artificial chromosome. So then the question is, does it make chromatin? And we're going to find out that yes, it actually generates chromatin, which winds up into chromosomes. So chromatin is DNA plus proteins. The proteins are provided by the yeast cell. They bind to this artificial chromosome, and they do function in the same way that they function for other chromosomes in the cell. So that's interesting in itself. We'll take a closer look at that in just a moment. They also looked at transcription. Transcription is the action of reading the DNA and pulling out a copy of the DNA and making it into RNA, ribonucleic acid. And the ribonucleic acid is the message that is sent out of the nucleus into the rest of the cell, where it will find the ribosomes, which are the machine that can read the code and turn it into proteins. they show that, yes, it is transcribed. This random sequence is transcribed. They can find the transcripts of portions of their random sequence in the yeast cell. All right, so that means there's the machinery, right? A very large complex protein structure, which includes something called RNA polymerase, is actively working and reading, all right, at the DNA level. They're reading or copying the DNA code into RNA code and sending it out into the cytoplasm of the yeast cell, where potentially it could be read into proteins. Right? Then they actually look at different forms of transcripts, and they find a wide variety of different forms of transcripts. And the bottom line here is that almost all of those 18,000 base pairs are transcribed in the cell into RNA. Let's look at the in brief abstract here. Biological noise. Interesting term. You may not have heard of biological noise. What is biological noise? It's defined as functionally insignificant events that occur in living cells due to imperfect fidelity of biological processes. As much as we'd like to think that we have this fine-tuned chemistry in us, where every single reaction is completely controlled, there are a number of chemical reactions that are happening to us that we don't have much control of, but they happen at fairly low levels, and this is called biological noise. And they're considered to be insignificant events because they're not chemical reactions that are occurring at a great enough frequency that they would have any impact, significant impact on the function of our cells, right? Or on our phenotypes, the way we look and act and integrate things in our bodies. Right? By analyzing chromatin structure and transcription of a randomly generated sequence of yeast cells, the authors here show that the transcriptional noise occurs at levels compared to typical genes. So if we were to take a look at your chromosome number 1, and we were to ask across that all those hundreds of millions of base pairs, you have certain places where there are genes, and those genes are turned on or turned off. They may not actually be completely turned off. They might have some very low level of transcription in which they're being copied, maybe even being translated in the cell, but maybe not high enough levels that it matters at all, right? We call that kind of like leakage, really, right? Or the term here, biological noise. It's just background noise. You're doing a little bit of chemical process. You can't completely shut off some things, but then you can turn up the spigot, right? And you can enhance a chemical reaction, or you can enhance a gene being promoted and therefore turn it on at higher levels such that you make a biologically significant amount of that product, like biologically significant amount of insulin, such that it actually affects your physical processes. But it's more than just genes being off, on, but not really being quite off, and leaky genes. We're talking about other areas between genes. They are a lot of times transcribed. A large portion of your genome is just at a very low level being transcribed. You have molecular machines that are looking for places to copy your DNA into RNA. And many of those places you don't really want to have copied because you can't really use that particular sequence. Some of it is, I hesitate to use the word junk, but it is sequence which doesn't actually perform a function. And yet you're going ahead and you're at very low levels actually copying some of that sequence into RNA. That's all biological noise. So if you look across your chromosomes, there's a certain amount of biological noise there, a certain amount of chemical reaction, chemical activity going on that is not part of your, like, who makes you what you are, right? It's not a significant event. And so when they put in this random sequence, you could predict that random sequence, because we would also predict that a lot of our sequence looks random in the sense that it doesn't have any perceived important function. So they made random sequence, which they know is random, right? You could always argue in a chromosome itself that maybe there is purpose to some sequence that we just simply haven't discovered yet. All right, there's a low level of transcription, and maybe under certain circumstances during development somewhere, we're actually using those transcripts for some actual actual thing, all right? Something that actually is important to us, right? In which case we'd say that's a functional part of our genome. We wouldn't want to get rid of that part of our genome, right? But the other hypothesis is that many of our portions of our genome really don't have any important function during our lives or the lives of any members of the particular species. And they're simply just being passed from one generation to a next, but they're legally, all right, they're creating biological noise as they are being copied. So by taking a look at this artificial chromosome, they can basically the hypothesis is that if we put in an artificial sequence, a random sequence, that it will also be wind up into chromosomes and it will be transcribed and it will form copies of these things, and it's trying to read it. In other words, it's biological. It'll create biological noise as well. And if it creates biological noise, that gives more weight to the idea that there are portions of our chromosome or of any other of the yeast chromosome that don't actually have a functional significant role for that organism. And the noise that comes from that is just that. It's just noise. It's biological noise. It's not significant. This plays an important role in some of the questions in which some people perceive that all sequences, right, must have some kind of important function for the cell, or maybe not important, but a function in the cell. And many scientists think that there are portions of the genome that simply don't have a function, right, other than simply taking up space, right, in the genome. So maybe some organizational structural reason, but not like actually making products. So let's read the summary, and then I'm just going to, I have one other figure to show you, and we'll just talk about these results a tiny bit more. We measured transcriptional noise in yeast by analyzing chromosome structure and transcription of an 18,000 base pair region of DNA whose sequence was randomly generated. Now here's the first finding. Nucleosomes fully occupy random sequence DNA. Although nucleosome-depleted regions are far less frequent, and there are fewer well-positioned nucleosomes and shorter nucleosome arrays. What in the world are they talking about there? Let me show you. Okay, so here's a figure showing, again, I was describing earlier that your chromosome is made up of proteins plus DNA. So here's your DNA strand, and your DNA strand then winds around a protein, a large complex protein called a histone. And the two together, the DNA plus histones, make a structure called a nucleosome, right? And more broadly, we're going to call this chromatin as we have these two things together, and you have one nucleosome after another. So in that abstract, they said that we put these 18,000 base pairs of sequence, just naked sequence, into the yeast. And then what should happen inside of a cell is that your DNA is not just naked DNA laying there, right? It's wound up around nucleosomes. It's a way of organizing your DNA. It's a way of structuring your DNA. Your DNA would get it would be a giant mess if you just had millions of base pairs of strands of DNA, right? They would get mixed up with each other, right? Creating crazy knots. And so what you do is you wind your DNA up around these histones, forming nucleosomes. And then the nucleosomes come in packages, and then you end up winding those up and so forth into even higher level structures, which form your chromosome. So you've got your nucleosomes. What happens to these random sequences? Well, first thing we learned is nucleosomes form. So that's just automatic assembly. That's self-assembly is happening here. They didn't have to do anything. They didn't give any instructions to the cell. They didn't make anything that's part of the DNA that would cause the nucleosomes to form. But the proteins, the histone proteins that are in the yeast cells, which are being made for the other chromosomes, they simply find and attach to this new piece of DNA that's been provided to the cell, and they wind the DNA up around those histones. There are certain sequences of DNA that allow for these attachments. And in the 18,000 base pairs of random sequence, there's going to be short pieces of sequence that happen to have the same type of sequence and charges and so forth that's going to make them wind up around histones. Now, what they did notice was it wasn't quite as structured. All right, your histones come in little packages that are fairly well organized in the sense that there's a histone and then a certain number of bases before the next histone and then a set of bases before the next histone and so forth. So you have sort of a regular pattern. What they found with the random sequence is they found a little less regular pattern. They found little clumps of histones and then maybe a larger section without histone and then more histones. So I don't think that that's surprising, but it may be surprising to you that it can self-assemble histones to begin with, and so it makes a chromatin structure, on the way to making a chromosome. Now, the thing about histones is that they shield the DNA from being able to be transcribed. So if you want to transcribe, that's what's being shown down here. You have the RNA polymerase. You have to have an open strand of DNA to look at. And so you have to unwind the DNA off the histone to reveal the strand of DNA. And then from there, you can open up that DNA and you can pull a piece of RNA out of it, right? That's the process of transcription. And so what they found in this paper is that, sure enough, transcription happens. In fact, transcription happens like all over the place. All kinds of little sections of this 18,000 base pairs are transcribed. That's because in random base pairs, you're going to have things like TATA boxes, right? TATA. Actually, it's a little slightly more complicated than that, but that's the typical name for a TATA box. So there's a certain sequence of code that allows for protein, all right, activators or transcription factors to bind to that, which then help the RNA polymerase, the protein, bind to that, which then allows for this process of opening up the DNA because it's double-stranded. You have to open it up and you'll be able to come in and be able to copy out the RNA from it. Right? In order to do that, you have to unwind the histones. And so the yeast comes with molecular machinery to do that. And so sure enough, the histones self-assemble on the random 18,000 base pairs. And then you have ribosomes come along and the various transcription factors, and you unwind DNA from the histones, and then you bind to that DNA because there's going to be sites that have short sequences that are randomly going to be, that are part of the random sequence. They're going to have accidentally found transcription start sites, right? If I just had you write out 1,000 base pairs of sequence, you would inadvertently write out several different transcription sites in that sequence. And so these protein complexes find those transcription start sites and they start transcribing and they make a transcript. And then that transcript can be transported out to the ribosome. Then the question is, does the ribosome actually read the transcript and try to read it into a protein? And they didn't go as far as that because they didn't do the protein part of this project. So they didn't look at all the proteins being made and try to line them up with these sequences. They're more interested in this first portion of the biological noise, because this is what's happening inside of you. All right. You have millions, right? Billions of base pairs of DNA. And many of those places where you have genes, you have various proteins and things like that they're clamping down on. Actually, you have histones that are resistant to being unwound, and that results in it being hard for you to find that gene and express it. And so you are intentionally turning off genes, all right? And we're pretty effective at it. And that was another difference that they found in this particular study. They found that that there's actually as much, if not more, noise occurring here, meaning there is more transcription occurring on the random sequences than there are on actual sequences of the yeast genome itself. And that's because, and again, I think we'd expect that, because the yeast genome is intentionally suppressing transcription in some portions of its genome. They are actively recruiting proteins and so forth to bind to the DNA to stop this process of opening it up and allowing it to be transcribed. And other places they're actively promoting, all right? They have transcription factors, what's called co-activators, that are telling the cell, I need this particular piece of information to be exposed and expressed so that I can use it to do some task. And so the yeast genome sequences are more like there's some basic noise across the whole genome because it's hard to stop every chemical reaction. You can make them really inefficient, but you can't completely stop it. But nonetheless, you can suppress those chemical reactions to very low levels. Whereas on the random sequence, they tended to have a slightly higher transcription rate. In other words, the biological noise was kind of, it was even all across the chromosome, the artificial chromosome, as opposed to high in some areas and low in some areas. So this isn't, we're not making any claim, these authors aren't making a claim that this random sequence suddenly became a functional chromosome in the sense of it is making products that are important for the survival of the yeast cell. No. What they're testing is this basic idea that the transcriptional apparatus, the histones, making chromatin, the self-assembly idea, that all these things can occur just if you throw in random sequence into an organism. Don't want to belabor this paper, so let me just read a little bit more of this abstract. Steady state levels of random sequence RNAs are comparable to yeast RNAs. Right, so that's what I was saying, that you have transcription occurring in the random sequences. But transcription and decay rates are higher. So the transcription rates are actually higher, again, because I was just saying that in the yeast, the regular yeast chromosomes, there are portions of the yeast chromosome, of the DNA, where there's suppression occurring, right? Intentional suppression of portions of the genome such that they are not copied. After all, it's a waste of energy to copy a sequence that you're not using. That's the thing about biological noise. The biological noise is copies of stuff you're making all over your genome, and you and I are doing it right now, right? You're making copies of lots of portions of your genome that are not going to be translated into proteins and made into anything. So you copy them, they make RNAs, and then they're degraded. You just recycle it and make it into something else again. That's essentially wasted energy, but you can't stop doing it because of the nature of chemical processes, right? It's the inefficiencies of the cell. It's the biological noise that occurs because, as they said up here, due to the imperfect fidelity of biological processes. Your enzymes aren't perfect, right? They don't always do the right chemical reaction. They do it at different rates. Your big machines that have to look and find locations in your genome in order to turn them on, they are looking for particular sequences, but they're sloppy. They're looking for like the ta-ta box, and that has a particular sequence that works most efficiently. But, you could have a section that has a C instead of an A, and most of the other bases are the same, and then the machine looks at it and goes, well, that's not perfect, but I can still grab onto it, and I can still do a little bit of copying. I'm not as efficient, so therefore I'm not making as many transcripts, right? So that's a site where maybe you didn't actually want to transcribe anything, but you had by chance the sequence there that's very similar. And so the lack of fidelity of biological processes results in you making things you don't need. So that's the biological noise. These observations indicate that transcriptional noise occurs at high levels in yeast. So the fact that they see the same types of things occurring in the random sequence as they do in the rest of the yeast genome suggests that a large portion of the yeast genome, which is being transcribed at some low level, is really just biological noise, right? So like, for example, the ENCODE project, which looked at functional elements in the human genome, one of the things they looked at was transcription. And what they discovered was that, yes, most of the human genome is actually being transcribed. And many people look at that and go, oh, well, most of it's being transcribed, so therefore it's functional. No, actually, it's just biological noise still. You have to take a few more steps in order to prove that it's actually functional, that it serves some purpose in the genome. And it's not just the product of the inefficiencies of chemical reactions, right? The lack of fidelity of our biological processes, right? So finding noise is not finding function. They provide insight into how chromatin and transcriptional patterns arise from the evolved yeast genome. And what's also interesting about this, and this goes to my other video in which I did 5,000 base pairs of random sequence. I looked for signatures of transcription start sites showing that sequence, if you took it and put it into an organism, I would expect would be transcribed. And here they've actually done it with 18,000 base pairs and shown that, yes, you actually get transcripts in real live cells, right? So I feel pretty confident in my predictions about the 5,000 base pairs, and I showed like a whole bunch of different places where I thought it would be transcribed. In fact, I predicted that most of those 5,000 base pairs would be transcribed into messenger RNA. Then what I did was I looked bioinformatically at those transcription sites, and I looked within those sequences, and I asked, are there start codons? And sure enough, there are start codons, ATGs. A start codon tells the ribosome where to begin translating and turning the transcript of RNA into the sequence of amino acids, which is a protein. And so I said I predicted that there were multiple sites in that 5,000 base pairs where there's a start codon and then there's a stop codon, which means there's the possibility that it can translate that section into a protein. So I think it would make proteins, and I would predict that these yeast are actually making some proteins from this random sequence at low levels, but nonetheless, they're making proteins in the cells. I feel pretty confident in saying that. Do those proteins do something? I think of highly unlikely, right? They're random proteins, random sets of amino acids that fold up into some sort of shape, but I'm sure the yeast doesn't have any idea what to do with those shapes and do those things. They're not making enzymes. They're not performing a function. Maybe they're sticking to something. Maybe they stick to another protein. If they do, they could alter its function. If they alter its function, they could actually alter one of the phenotypes of the yeast. It changes what the yeast is and what it can do. If that's the case, I'd call that a function. So it's possible that a random set of sequence gets transcribed, sure of that now, gets translated, pretty sure that happens, and then a few of those may actually interact with the cell itself, potentially changing what that cell does. And so you're seeing the process of how organisms can actually find new genes in random sequence. And most of the genome is random sequence. It looks like random sequence. It's just generating biological noise. But sometimes biological noise can find a use. If it finds a use, if it has some sort of phenotype that then gives some positive benefit to the organism, then potentially that will experience more mutations, which will then enhance its properties, making it more beneficial for the organism over time as natural selection acts upon it. And so this is just another example just published of how random DNA, you can see the path from random DNA to being made into genes that potentially serve functions in organisms over time. And we have many examples now of this whole thing of junk DNA actually turning into actual genes in organisms. And this is just a nice little snapshot of the beginnings of that process in this particular paper. All right, so functional analysis of random sequence chromosome reveals high level and the molecular nature of transcriptional noise in yeast cells. Thanks for listening. Hit the like button, do all those types of things. I really appreciate it. Talk to you later. Bye-bye.